is Yuki Kato from Osaka University, Japan. Our presentation title is Alignment of Complex Single-Cell Trajectories with Capital. First, let's take a look at an example where some tissue is obtained from a mouse. We can assume that it contains a heterogeneous population of cells, including undifferentiated cell and mature cell. That means a snapshot of something like time course data. After applying this to single-cell RNA-seq and obtaining a transcriptome profile, we can infer a cell state trajectory along the pseudotime. Then, expression dynamics of a gene can be investigated with respect to the pseudotime. Next, assume that we have two mice. One is a control and the other is a diseased model. Here you can see a cell state trajectory of each mouse, which shows two or more differentiation pathways. Now we try to compare differentiation 1 with differentiation 1' across two mice by aligning cells along the pseudotime. Then we can see, for example, this expression dynamics of a gene A. That implies that gene A would be a factor of disease. However, the problem is that how we can know the trajectories to be compared beforehand. So far, a few methods have been proposed to compare linear trajectories, where we may have already known the trajectories to be compared. On the other hand, we propose capital that can compare tree trajectories. That means we don't have to have prior knowledge of how to choose trajectories. That is also reasonable because cell lineage is known to form a tree. Details of the algorithm can be found in bioRxiv, which I will show in the final slide. Okay, let's move on to the evaluation of capital with two data. One is synthetic and the other is authentic data. First, we used DynGen to generate a pair of synthetic data, where the number of cells is 1000, the number of genes is also 1000, and the backbone structure is bifurcating, shown here. Figures on the left side are data 1 and data 2 ground truth trajectories. SA and SB are the names of states. These on the right side are predicted clusters together with trajectories with capital. Each state was annotated by looking into the expression of a marker gene. In the ground truth, SB mid bifurcates into SC and SD, or in the prediction, SC or SD bifurcates. That would suggest that it was hard to classify three intermediate states. After building a trajectory of each data, capital can compute a cluster alignment. In the next slide, we will focus on this linear trajectory. Let's take a look at the cell alignment result for simulated gene B3TF, which was computed by dynamic time warping. The reason why we focus on this gene is that it can differentiate the intermediate state SB mid. As you can see, the orange cells in data 1 were aligned with the orange ones in data 2. And the green cells in data 1 were also aligned with the orange ones in data 2. That would suggest that the green cluster in data 1 will have a similar profile to SB mid, which bifurcates in the ground truth. Next, take a look at gene B3TF, indicated by a green curve. The feature of this gene is that expression decreases dramatically when the state moves from SB to SB mid in both data. That can be correctly predicted with capital along the pseudo time. Okay, let's go on to the evaluation of capital on human bone marrow cells, which were obtained from Seti's paper and Welten's paper. Figures on the left side are predicted clusters together with trajectories with capital. After aligning these clusters, the figure on the right side was obtained, where yellow shaded part is almost correct alignment. However, some matching clusters were wrong, as the inferred trajectory was not accurate, as shown here, for example. Finally, I have to tell you, details of the algorithm can be found in bioRxiv. Also, capital Python codes are available at GitHub. As a future work, we need to investigate the possibility of enhancing predictive performance of pre-capital, meaning before alignment. Thank you for your kind attention.